So welcome for this afternoon's session, which will have several talks, and we'll try to go towards both the role of genomes in primary prevention, try to identify the functional variation in human genomes, and also the last one, and Nikos is not yet with us, he's actually probably waking up slowly because he's in the US, on how you go and try to do drug discovery in the post-genomic era. The first speaker of this afternoon's session is Samuli Ripatti, who is actually coming from the University of Helsinki. He's trained as a statistician and professor of biometry at the University of Helsinki. He's vice director of the FIN, which is the Finnish Institute for Molecular Medicine, and director of the Center of Complex an excellence center of complex disease genetics. I guess he's going to talk to us about how the beauty of statistics and genetics and how they have to be played around, because being a trained geneticist will be his prime interest, but also how this is used for a better health, a better understanding of the genomes, and a better understanding of how we can live a better life in the long run if we are not only Finnish, but also European at large. So Samuli, the floor is yours.
Thank you very much for the kind introduction and for the opportunity to show some of our work on this domain. So what I will talk about is measuring genetic risk and the sort of the downstream applications that we can potentially have for doing that. And the tool, since you mentioned statistics, let's go straight to the statistics here. So the tool that has been become pretty well established is measuring not only the genetic risk related to individual variants in the genome, but rather trying to capture as much as possible of the risk along the whole genome. And the way to do this is really putting together our information on the role of each variant that we capture, typically in large-scale genome-wide association studies, either filter or model the correlation structure between the variants, the LD structure there, and then squeeze this information into a measure of individuals' own genetic risk. And out from this type of an exercise, we typically get a distribution where each of us are somewhere along the line of this one-dimensional measure. And then we can start correlating this risk factor, genetic risk factor called polygenic risk score, to actual disease probabilities or longitudinal probabilities of the risk. So this is kind of in the background of pretty much what I will present today. And I'll show a couple of examples, couple of open questions related to this type of approach that we need to still solve before we are in a position where we can start utilizing these in clinical practice. And different disease areas or diseases are in a little bit different place in relation to this. So overall, why has this type of tool, polygenic risk scores, become so successful or so widely used is due to three things that have come together. First of all, we are now, because of the large-scale genome-wide association studies, we are right now in a position where we can estimate pretty well the contribution for each individual variant to the risk of a disease. Second of all, the algorithmic development related to capturing the risk has become pretty good. I mean, we started out with very few variants in this game, but now we are modeling the whole genome variation. And a third component is the availability of large-scale biobanks and large-scale cohort studies where we can test these models, or train and test these models around. Our biobank or biobank study that I use most of what I'm going to present here is the FinGen study in Finland, where our aim is to collect 500,000 Finns. 500,000 Finns is a different thing than 500,000 UK people in the sense that this is really 10% of the population. So it's a big national push to to put this data together, everybody is linked to our Nordic-style National Health Register data. Some of the registers go back to the 1960s, so that we have actually, for example, for the hospitalization data, we have 50-year follow-up for everybody in the database, as well as, say, every prescription drug purchase we have since 1995 captured there. We developed a version of the Axiom GWAS array that was Finland specific, and then we combine these sources of information for everybody and then do downstream analysis. This is of course used very much in the FinGen fashion, first of all to run systematical GWAS studies for the data to discover novel associations, and particularly among the coding variants that are slightly deleterious but because of our population history are enriched in Finland. This is sort of the promise of FinGen, and sort of it's really a public-private partnership, in the sense that in the background there is both Finnish public funding agency with a big funding part, and then a sort of pre-competitive setting. In a pre-competitive setting, we have now 11 pharma partners in FinGen. So this is kind of in the background. So this is the data in a very short introduction that I'm going to show or utilize for. So one of the things that we have been doing is simply follow my first slides protocol and tried out how well are we now able to capture the cumulative incidence or cumulative disease rate for different diseases. Here you see 5 different diseases: coronary heart disease, type 2 diabetes, atrial fibrillation, and breast and prostate cancers on the second row. But I think overall what you see here is that the risk itself, although sort of the age dependency of the curves differ, as we know, these diseases come a little bit in a different part of life, but you can see that the distribution or the variability in the risk distributions are pretty much similar, regardless of which one of the 5 plots you see here. So this is in the background. Now we know that for many diseases, those ones particularly where in the background there is big enough genome-wide association studies to give us the weights for individual SNPs, we do capture very well the variability in risks. And now the question are, what are the next things to look at after this, if we see this? So I go through 4 key questions and answers that sort of prevent us right now jumping into the utilization of these tools in clinical setting. So the first immediate question is that for many diseases we have already routine clinical risk tools. So the question is, how much does the polygenic risk score provide additional value, or does it? Is it so that this type of risk is actually already captured when we measure our cholesterol levels or other biomarkers or blood pressure levels? Second question, for many diseases we actually know and test routinely high impact mutations. So another question is, if we align these together in diseases such as breast cancer, how do they align with each other? Does the PRS capture the same mutations, or is it additional information? Third question, why not simply measure genetic risk by asking about family history of the diseases? That's of course something which is very simple question. And why bother with all of these types of tools? This is a question that I get often when I give these types of talks. And then the fourth one is that, can we use polygenic risk estimates similarly in all ancestry groups? So I think these are some of the very fundamental key questions that we need to answer. And I hope to go through these over the next 20 minutes or so. So this is the state of the art in terms of clinical risk utility for cardiovascular disease. This is SCORE, the European fatal CVD tool or clinical risk score. But there are tens of others, but very similar variables go into these calculations. We have sex, we have age, we have blood pressure, we have smoking status, we have cholesterol levels, and these then have in different calculators a little bit different weights, but that's pretty much it. What you see here, if you're in the red, it's bad. If you're in the green, it's good. But what you can also see here, that if you're a 40-something woman, regardless of what your values are, the score predicts that you are in the low risk group. So you can smoke and you can have a very high blood pressure and so forth, and it really doesn't make a lot of difference. Similarly, if you're a male over 65, regardless of how good your values are, you're always in the very red end of the scale. So in some sense, these tools that often look at the risk over the next 10 years are actually not very valuable, except for a sort of a relatively short range of lifespan for individuals. There are also more advanced versions of it here. We have a Dundee coronary risk disk where you circle the disk, and it's a nice mechanical risk calculator, essentially doing the same job there. So for coronary heart disease, if we zoom into a little bit on that, in parallel, of course, over the last 10-15 years, we've learned a lot about the genetic background, which is not really taken into account in the current clinical tools, or it's only taken into account by measuring some partly heritable risk factors. So we know genetic variation explains about half of the disease risk variation between us. There's close to 100 genetic loci now associated with CUD risk, actually, and now the next wave of genome-wide association study, that's going to be at least doubled, if not tripled, that number. There's been the discussion of if we take a coronary artery disease patient and think that there's genetic background, the question is if it's driven by individual high impact mutation, or if it's driven by a blend of causal risk factors. And what I'm talking about today is mostly on the latter one type of approach, but I also contrast these two a little bit in the context of breast cancer. And of course, if we want to know individuals' risk, we need to be aware of both. So genetic risk of coronary artery disease, the development of polygenic risk scores over the past 10 years. We started out in 2010 with what we thought was a monster of a polygenic risk score with full 12 SNPs calculated, being input for the score. And then over the 10 years, we sort of become more and more aware that there's so much information under the genome-wide association study peaks that can be utilized for the risk scoring that now we more or less try to take in as much as possible and model the LD along the lines. So if we go to the UK Biobank European Ancestry Set, calculate the polygenic risk score, look at the top 1% of the score, it's about 5 times odds ratio between the top 1% and the rest 99% of the distribution, which is relatively big odds ratio. So in a sense, in these types of settings, in UK Biobank at least, there seems to be a lot of information captured by this. The question, again, from implementation, from primary prevention point of view, is that how does the PRS work with the current clinical risk scores? So just to remind you, so for coronary heart disease, when we look at the top 2.5% of the distribution on red here, we can see that for those individuals, the lifetime risk of coronary heart disease is about 65%. And if we go to the protective end of the polygenic risk score, it's about 24%. So this is the spread that we capture with the current tools The question, how does that align with the clinical risk score? So here we have divided the FinGen cases for the 5 different diseases into 2. On the top line, we have early onset cases. So those cases, a little bit depending on the disease. Again, there's the same 5 diseases: coronary heart disease, type 2 diabetes, atrial fibrillation, breast cancer, and prostate cancer. You can see that so we asked the question, how often in the individual who captured the disease, how often is the clinical risk scores high so high being defined as based on the guidelines, and on the red we see how often is the polygenic risk score high up, and on purple are the individuals or the cases where both scores were high. So you can see that for the early onset cases, there is actually a lot of, if we play the game, would we have identified early on this individual who got the disease early in life? If we had the information on the genetic score, how often would we have identified it? And for it varies a little bit from one disease to another, somewhere between 10 and 30% of the early onset cases we would have actually identified if we had the genetic information on. When we go to the bottom row, there's a lot more blue there, meaning that for late onset cases, we are much better with the clinical scores, and the additional value that the genetic score brings is there for type 2 diabetes and breast cancer, clearly, maybe for prostate cancer as well, but not as much for coronary heart disease. So we are good at identifying with clinical scores both atrial fibrillation and coronary heart disease. So in other words, it depends on a little bit of the when the disease onset is, in terms of what are the relative impacts of the clinical score and high PRS. But overall, in the distribution here again, coronary heart disease on the upper plot and type 2 diabetes on the bottom, you can see that they are more or less additive or multiplicative on the risk scale, but meaning that they bring additional value to each other. So measuring both is much more valuable than measuring one or the other only. So I think this is one of the key messages when we are going to implement them. Often you see the genetics community talking about polygenic score and the clinical community talking about clinical risk factors, and these two communities that do not necessarily talk to each other, but we should actually talk. They should talk to each other and bring all of this information together. You can actually sort of pose this same question the other way around. Now that we have tens of millions of individuals who have been genetically profiled, either in large-scale biobanks or direct-to-consumer companies, how could we utilize those data for prediction purposes? And we asked the question, if we have the genome information, if we have age and sex and PRS available, what additional value do the easily available risk factors provide for that? And so on the left-hand side, coronary heart disease, if we simply ask from the individuals about their smoking status, BMI, the usage of antihypertensive medication, their diabetes status, family history of coronary heart disease, and these questionnaire-based, simply, you can have a very simple web form to collect this information. You pull this information together, add it on top of the PRS, we can actually get the GRID CHD score in there. Oops. And you can see that the AUC for GRID CHD line there is on the same level as in the bottom with the two routinely used clinical scores. So simply by adding a handful of questions on top of your PRS, we can actually get a very simple tool for, at least for initial check of the the person's risk. And then if you add on top of that systolic blood pressure, LDL and HDL levels, you can actually do much better than the routine clinical scores. Now, this is a model that has been trained with FinGen data, and now these results come from UK Biobank. So it's an outside sample check for that. Okay, question number 2. How does PRS work with well-known high impact variants? Here I go now to example breast cancer. So if we think in terms of what we know about breast cancer and what we routinely test in our clinical panels, it's the rare high impact variants, as well as some of the low frequency variants with moderate impact, such as CHEK2 and ATM. And if you contrast them, the allele frequency on the x-axis against the effect size on the y-axis, the common variants identified, there's about 250 common variants identified as well on the line. The common variants, as in many other diseases, have much smaller effects. But when we start putting them together into these scores, suddenly we have also in breast cancer, we have a comparable tool to measure the risk with some additional properties. So one of the questions that we've been looking at is that where should we, if we look at the polygenic risk score for breast cancer, where do we go if we want to have a comparable risk to the well-known high impact mutations? PALB2, an example here for a mutation that gets you over 40% lifetime risk for breast cancer if you carry. This is a mutation that is, by the way, highly enriched in Finland. So it's about 0.25 of the top distribution of the polygenic score that gets you roughly to the same scale, and the top 10% gets you on the PRS distribution, gets you to the same risk as some of the moderate level mutations, such as frameshift mutations in JET2. So if we ask the other way around now, if we go for a random breast cancer patient and ask how often do we see the mutation, high risk mutation, we see about 1.5 women out of 100 carry in our data sets those high risk mutations. The high polygenic risk, comparable high polygenic risk is for 1 woman in 100. 5 women carry moderate risk mutations, and 21 actually have polygenic risk, which is on the same level. So on the population level, the polygenic risk has much or touches much larger number of women than those high risk mutations that we at least have cataloged so far. Does it matter in terms of the additional risk for that the polygenic risks brings you if you have a mutation or not? Short answer, it doesn't matter. These are independent of each other, regardless of whether you're carrying the mutation or not. The distribution at the additional risk in the high end of the polygenic risk score is the same, and the protective effect on the lower end of the polygenic risk is also the same. So, which means again that we should look at both of them. So it's not that one captures the other. So this is what we did with Nina Mars, is that we compared the cumulative distribution of lifetime risk. On gray, you see the population level by the age of 80, it's about 16%. If you're a carrier of CHEK2 frameshift mutation, Finnish enriched mutation, it's about 30%. If you're a carrier of a CHEK2 mutation and you're at the top 10% of the polygenic risk score, it goes all the way up to 60% cumulative incidence by the age of 80. On the other hand, even if you're carrying the mutation, if you have a lot of protective polygenic risk, that actually brings you down to the population level of risk. in terms of lifetime risk. In other words, when we talk about the penetrance of a mutation, this is one reason why the penetrance is, of course, different, is that it's not only single mutations that operate for the risk, but rather a combination of mutations and the long tail of risk variants for polygenic risk. We could play the game that should we apply differential risk evaluation when we invite, in Finland, there's a nationwide system for mammography screening. Every woman, when they turn 50, are called for biannual checkups until 69. The sort of rationale is that by the age of 50, there's 2% breast cancer prevalence in Finnish women. And when we look at our data, FinGen data, we see that the middle part of the polygenic risk distribution hits very nicely the same age. So by the age of 50 in our data, also the breast cancer prevalence in the mid-range of the polygenic risk score is 2%. When we go to the higher end of the risk distribution, the same 2% prevalence comes about 5-6 years earlier for those women. And if you have a lot of protective, and you can, it's actually 8 years later. So these are now inputs for the sort of the cost-benefit analysis for whether we should start applying more risk-based strategies for screening for breast cancer. Okay. Then my slightly provocative question: why not simply measure genetic risk by asking about family history of the diseases? There's of course sort of good things on the green here about family history measurement. It captures at least partially genetic and lifestyle factors. It's cheap, it's easy to measure, but then there's obvious problems with it. I'm not sure how well we know what our aunts or uncles have, what diseases they've had, or it's sensitive to how these are asked and so forth. So and we actually need to have the disease actually manifested. So if it's different if you're 5 brothers and you are the oldest versus youngest, it's going to be a different answer to this type of question. So when we look at FinGen across 27 diseases on what is the what is the additional risk when we compare the first degree relatives. I mean, when we are covering 10% of the population, we tend to have a lot of actually family members in the data set, not necessarily by design, but by chance as well. So first degree, we have first degree family history, second degree family history, and parental causes of death there measured for 27 diseases, and you can see that overall you see that for many diseases, the first degree, if you have a positive family history for a first degree relative, you have about on average sort of on the level of 2 plus a little bit more additional risk. This is for breast cancer, 2.36. Here's for coronary artery disease, it's about 1.6 is the odds ratio in our data set for first degree relative having the disease. On average, the mean effect size is about 2.2. If we go to the second degree family history, it's about 67% lower, 33% lower when we ask the question, do your parents have they died for the same disease, for those diseases where it's relevant to ask that. So in other words, family history does matter. So then the question is, if we adjust for, here is plotted the same risks for the same diseases for the being at the top 10% of the polygenic risk score, and you can see prostate cancer leading the way there. If you're at the top 10% in the prostate cancer polygenic risk score, it's about 4.5 times odds is the odds ratio. The second, the sort of the on the right for each estimate, there is the same estimate for the top 10% of PRS adjusted for family history. And what you can read from here is that there's a very little change in the estimates. In other words, the family history not capturing your individual genome that much, but rather if you adjust for that, you get roughly the same estimate. The mean decrease is 3.4% in the PRS effect sizes when adjusting for first degree. If you ask the question another way around, does PRS explain away the first degree family history information? It doesn't either. There's a little bit of more adjustment effect, but it's again the same thing. These both are important risk factors, but they do not cancel each other out. So, and there's no systematic evidence for interaction. So why is there so little overlap between the effects of family history and PRS? And this is we have scratched our head a little bit, but it's a very clear result that family history, of course, measures both shared genetics and environment as well. So it's not only genetics. For family history, a similar risk is assigned to all relatives of the same degree. If you have 5 siblings, they really don't have the same risk, regardless of if their father has, say, died out of coronary heart disease. And you need to have you need to be in the phase of life that you have the family with the disease, or possibility to get the disease as well. And the genetic side, of course, is your individual realization of risk in the sense. So this is sort of the contrasting of the two. Final point: does ancestry matter in these types of results? So my results came from Finnish data sets. Does it matter if we go outside of the European ancestry groups for 4 diseases? And what you see here is on green is the odds ratio for 1 standard deviation change in the polygenic risk score. On blue or purple, there is Asian samples, on red, African ancestry groups. What you can see is that Asian here meaning South Asian plus East Asians together to get the necessary power here. So what you see here is that Asian samples have almost the same level of risk captured by as European ancestry, but African ancestry groups has much less association, meaning that these current polygenic risk scores really behave very lousily in African ancestry groups. Particularly if you look at breast cancer, there's no value at all for the polygenic risk score that I showed previously for African ancestry samples. If we zoom into Europe, look at different biobanks in Europe, you can see that they, regardless of which type of healthcare system they come from, or the ancestry sort of local ancestry backgrounds, they do behave roughly similarly. So within European ancestry groups, they work very similarly. So to wrap it up, in summary, about my points, it does make sense to combine genetic risk with clinical risk factors when available, not look one or the other alone. It does make sense to combine polygenic risk with simple health questions to provide comparable prediction to routine clinically used risk scores. We need to forget the idea that family history is a very good surrogate for genetic risk. It's a valuable risk factor, but it does not capture the individual genetic risk very well. And we definitely need to run more GWASs and do better algorithmic development to capture the non-European ancestry, particularly in Africans, to be able to estimate the risks for everyone. And with these words, let me thank a couple of key individuals. Nina Mars is a wonderful postdoc who's behind of much of the polygenic risk score work that I presented. Elizabeth Vidén is my partner in crime for much of the clinical translation side of these things, and Max Tamlander has been working with the questionnaire-based risk evaluations. Arno Palotie and Mark Daly are the running the FinGen show, and of course, without the FinGen, none of what I presented would have been, I would have been able to present. And with these words, I'm very happy to take questions or comments.
Thank you, Samuli. Thanks for this presentation. I open the floor for the question and answer. You have a button on the bottom right that you could fill. And if there are questions also from the panelists or the people, we have actually their video, just chip in. I see that Manolis is around, so you might want to ask a few questions. Is there any questions from the audience? Or from the panelists? Marc?
Maybe if I may. So, Samuli, you have data that show that we may now enter a fantastic time of genomic medicine for common disorders. And you also mentioned some kind of inertia from the clinical community. So I suspect that the clinical community would anticipate that you would need much more demonstration in translational study of effect on outcome and so on. But maybe there's a shortcut, as you mentioned. Why not go empirically, as we always did in medicine? So my question is, how do you think it should now be translated into clinical practice?
So that's a big question, of course, and it's in the back of our minds every day. And I think what we have been trying to do on that front, and of course it's a little bit different for different diseases. I give you two examples of the current translational ongoing studies that we are running. One of them is that for primary prevention, it's kind of a no-brainer in a sense to have one additional new risk factor to screen for, because the polygenic risk score really behaves similarly as your cholesterol measurement in terms of as an individual risk factor. So why not take that into account, particularly in settings such as Estonia is a wonderful example in the sense that they have the biobank, which is very much linked to the healthcare setting, and more so in the next phases. So there's a sort of a large proportion of population already screened for these genetic tools. Now it's a question of how to take them into account. When we have so we need tools to communicate these risks. When we've tried them out both in Finland and in Estonia, it's actually the clinicians are very excited about the sort of the opportunity to take these into account. So then comes, of course, the national guidelines, the European guidelines. That's one way of pushing these forward. I think what's going to happen is that we tend to be in Europe a little bit more conservative compared to our friends in the US. So it may be that the US will be the first ones to implement them in primary prevention at least. Another example comes from, since we've been working with breast cancer now a lot, is the fact that, again, that's a setting where there is an existing pathway of utilizing genetic information. We screen the mutation, we screen the BRCA mutations or CHEK2 or PALB2 mutations already at this point for early onset breast cancers, and we go to the healthy family members to screen for these. So putting polygenic risk alongside with these, now that we show that, we and others show that the risk is comparable. So since there is infrastructure already in place, I think there's sort of it's easier to implement these in these settings where it's just another tool on the toolbox. So I think these are, and once it's been shown in these couple of examples, then maybe the other disease areas will follow. But of course, you need to have a disease where you can do something about the risk. Of course,
first step is always the most
matter otherwise.
Yeah, first step being always the first one to take. I mean, it takes a long journey, but the first step is the most important one. We have a question from Zoltan Kutalik. So great talk, Samuli. Why do you think PRS contributing more for the early onset stratum compared to looking at the late onset cases?
Now, it does seem to, for example, for breast cancer, it does seem to be the case. So of course, the one potential explanation is that as we grow older, the environment kicks in more and more, and the genes are there already from the beginning. If you're at the high end of the genetic load, then you simply get the disease earlier. But it doesn't seem to be the case for at least all of the diseases that we've seen. And I think prostate cancer is a little bit different in terms of how the age, the relationship between age and the polygenic risk score is not as pronounced, at least there. It may be that because the onset is so much later that it sort of operates differently. But definitely, I think the environment, we let the more environment in as we go along.
Okay. Other questions? I perhaps have one actually. There is quite a lot. You treat disease in a separate manner, but we are all polymorbid the more we age. So how much the PRS could help track that polymorbidity or comorbidities, and sometimes the anti-correlation between these morbidities that exist. So
I don't think it has been looked at carefully. So I think the next wave of algorithms are going to be, I mean, now it's sort of natural that we start somewhere. We have relatively traditional definitions of the diseases, but I think there's more we could do by combining evidence, not only, so evidence between, say take type 2 diabetes and coronary heart disease. It's clear that there's information about your coronary heart disease risk in the type 2 diabetes polygenic score, which is probably not captured all by our current coronary heart disease score. So finding ways to pull this information, taking lipid scores, taking blood pressure scores into a more comprehensive genetic risk evaluation is probably something that we are going to see in the next wave.
Okay. So more work for the statistician to disentangle confounding factors, probably. Thank you very much, Samuli, for this presentation. We're going to move now to the next presentation from someone that is just next door, but originally coming from Finland. That's Tuuli Lappalainen. Tuuli received a PhD from the University of Helsinki in Finland. She followed that by moving to a postdoctoral fellow in Switzerland, in Geneva. We are all with our own Analyst, as we called him. And she then moved to Stanford while trying to beaver on the paper that was sort of accepted but needed to be reviewed. And then in 2014, she moved to the New York Genome Center, which was something that was pushed heavily by Tom Maniatis and a lot of other people in the old days, because New York had to have a New York Genome Center, like Geneva has to have a Genome Center somehow. In 2021, she was appointed as a full professor at the KTH Roller Institute for Technology, which is for the Swiss people, the EPFL ETH domain, and SciLife Lab, which is one of the pretty beacon in Sweden, where it merged technology, science, and research. And I think this is a great environment. She's one of the driving force behind the GTEx consortium. There has been a slew of paper, and this is probably a good way to transition from the genetics in the DNA to the function of it through QTLs. Without further ado, Tuuli, we are very happy to have you here and looking forward to your presentation.
Thanks very much for the introduction and for the invitation. And it's exciting to be back in the European research community after many years in the US. Still be going a little bit back and forth, but yeah, fun to be here. So I'll be talking about some of the recent work that my lab, in collaboration with many others, has been doing on interpreting the genome, sort of like thinking about what Samuli was talking about, kind of looking a little bit under the cellular hood of how do these variants actually end up affecting genome function and then ultimately contributing to disease risk. And this is, of course, an area that has long been recognized as one of the big challenges in human genetics. While we have these massive GWASs linking genetic variants to human traits, the functional interpretation or the functional mechanisms that mediate this link between teeny tiny genetic perturbations and very complex human traits is much less obscure. And one of the key approaches here has been linking genetic variants to molecular traits such as gene expression and other traits that we can measure in cells and tissues. And this has been particularly important and interesting for interpretation of CHUV loci, as we have known for a long time that the CHUV signal derives about 90% from the non-coding regions of the genome, where the function is not sort of readily interpretable from the genome sequence alone. And then there's also a lot of sort of like the generalizable information about the functional genetic architecture of human traits, and also interpretation of the personal genome. I'm not going to talk about that today, though. But actually, when we think about the sort of functional effects of genetic variants at the molecular and cellular level, there are actually many questions that we need to answer to fully kind of address this question. And luckily also many approaches to tackle these questions of what are actually the causal variants in, let's say, a CHUV locus of that kind of bundle of variants in strong LD. What are the proximal molecular effects of those variants? For example, do they affect transcription factor binding, enhancer activity, and things like that? What are the target genes in cis? So kind of in the local genetic space, what do these variants affect? What are then the downstream consequences in terms of regulatory pathways and cellular phenotypes that actually then ultimately lead to a physiological phenotype? And then what is the cell type, cell state context where these effects take place? And I'm not going to discuss all of these things that are on the slide on detail, but just to kind of like highlight that there are many sort of approaches that utilize the CHUV summary statistics, large molecular data sets from sort of population scale molecular assays, and many experimental tools as well to tackle these questions. And my own research has been kind of focusing on this type of layer where we take a lot of genomes, a lot of RNA-seq data, and put those together to map these regulatory variants. And I'll talk a little bit about that. We've also done a little bit of work in sort of functional GWAS kind of integration of those data sets. And I'll finish this talk by describing some new work from my lab and collaborators using scalable experimental tools to address these questions. So, but going back first, or starting from the kind of scalable molecular assays to map genetic variants and their effects on gene expression and splicing and other traits of the transcriptome. The largest effort in this space is the genotype tissue expression, or GTEx study, which was launched about 10 years ago. This is a big NIH-funded consortium that really looks at these questions across a diverse set of human tissues from postmortem donors. And there's been multiple sets of papers published from the different stages of the consortium, with the final set of GTEx papers being published about 6 months ago. And now kind of GTEx is basically coming to an end. It was an honor to be one of the leaders of the final stage of GTEx. And I've been involved in this project ever since I was a young postdoc in Manolis's lab. And he came to me one day asking that there is this consortium meeting in the US. I can go. Do you want to go? And I said, sure, I can go. What's this consortium? And that was how I got involved in GTEx. So in the final phase of GTEx, there is a massive amount of data and a massive amount of kind of discoveries coming from that. So altogether, we've been working with over 17,000 tissue samples collected from over 800 donors from 54 tissues from those donors. Not every tissue from every donor, but that's the full size of the data set. And then we have RNA sequencing data from all the tissue samples and whole genome sequencing from each of the individuals. And this allows us to map genetic regulatory variants we call expression QTLs and splicing QTLs that are simply genetic associations for molecular kind of quantifications, such as gene expression levels or kind of splicing patterns. So there's a little example of an eQTL in the middle. And when we apply this approach in GTEx, focusing on cis eQTLs and splicing QTLs here, there is more in the paper that are mapping sort of proximal allelic associations between variants that are close to a given gene. we find tens of thousands of these associations. So for the vast majority of human genes, we find common variants that associate to that gene's expression or splicing. And this is kind of, we see consistent patterns of this across different tissues here shown in the different colors, with the number of discoveries, of course, being largely driven by the sample size and statistical power. GTEx being a multi-tissue data set, of course, brings out the question of how are these effects shared between different tissues. So obviously the germline genome is the same in all of our cells and tissues, but the function of the genome and the effects of the regulatory variants in those different tissues are not necessarily the same. And we actually see the tissues cluster in kind of in a manner that makes sense. Kind of things that you think should be related are related. Brain subtissues cluster closely together, but actually there is a pretty high degree of sharing that was perhaps a bit of a surprise or something that I personally didn't necessarily expect, that between many tissues that are kind of seemingly not super related to each other, there's a very, very high correlation of regulatory variant effects. There are some notable exceptions, such as whole blood, that is not like a super great proxy for most other kind of internal organs and tissues. It's a relatively specific tissue type. But one of the novel things that we pursued in this stage of GTEx was analysis of cell type composition of these tissue samples. So when GTEx study design was put together 10 years ago, there was no such thing as kind of single cell sequencing or large scale sort of analysis of cell type composition. But now, of course, we acknowledge that this is an important component of tissues being composed of different cell types, and this being these being the core biological unit of kind of molecular function. So here we address this question by a computational deconvolution approach, where we estimated the enrichment of 64 reference cell types in GTEx based on the expression signatures of the tissue samples that we had in GTEx. And this plot just shows the simple kind of average enrichment of different cell types in different tissues. We see a lot of things that make sense. These approaches are not perfect, but they do kind of work and actually allow us to get this kind of population view into cell type composition. And one remarkable thing that came out of analysis of these cell type composition patterns is that this is a heat map of pairwise tissue sharing of cell type composition. And if you look deeper into this heat map, you can see that it is actually extremely similar to the heat map of eQTL effect size sharing between tissues that I showed on the previous slide. They're about 90% correlated, which shows that a lot of the sort of tissue sharing of genetic regulatory effects is actually driven by sharing of cell types between different tissues, which kind of makes biological sense that if you have, let's say, regulatory variant that has a very strong specific effect in epithelial cells, it would probably show up in all kinds of tissues that have a high proportion of epithelial cells. But it really highlights the fact that understanding cell type composition is going to be a really key factor in understanding biology of human tissues and the effects of genetic regulatory variants as well. In GTEx, we were able to use that cell type composition and actually its variation across different individuals for a given tissue to our benefit, in the sense that we were able to use it to map cell type interacting QTLs. And the way this analysis works is that kind of for any given tissue, you can do eQTL mapping and splicing QTL mapping so that you include cell type composition as an interaction term, and then you can map these kinds of situations, such as here, where so this is an eQTL for this particular gene in skin, and one can see that individuals that have a high enrichment of keratinocytes in these skin samples, they have a higher effect size of this eQTL compared to individuals that have fewer keratinocytes. And this is a signal of this regulatory variant being active in keratinocytes or cell types positively correlated with keratinocytes in a somewhat cell type specific manner. So we cannot necessarily assign the kind of, these are not cell type specific QTLs per se, but we can show that these are regulatory variants whose activity has cell type specificity. And doing this across 7 different cell types and multiple different tissues, we were able to map sometimes over 1,000 cell type interacting QTLs per tissue, and really expanding the GTEx resource in this way. Projects such as Human Cell Atlas that are, of course, doing fantastic deep work analyzing cell type composition of different tissues, they don't have the population aspect. And to be able to truly do this type of analysis in kind of using single cell RNA sequencing would really require kind of like the Human Cell Atlas times like 900 individuals that we had in GTEx to enable the genetic mapping. And GTEx also provides interesting insights into functional mechanisms of GWAS loci, which is, of course, one of the early major motivations of pursuing a project like GTEx to begin with. There is a very strong genome-wide enrichment of GWAS signal in QTLs, both in cis-E QTLs and in splicing QTLs, in such a way that when we kind of like basically hone in closer and closer to the likely causal regulatory variants, as opposed to the large sort of association signal of linkage disequilibrium proxies, we see a stronger enrichment, which together with other sort of similar indications really shows that these overlaps are not spurious, but there is truly kind of, these are causally mediating GWAS associations. There has also been both in GTEx and in other projects, many attempts to form hypotheses of causal genes for individual GWAS loci using approaches such as TWAS, predict scan approaches, and co-localization, where you basically try to find situations where your GWAS association signal in a given locus and your QTL association signal line up in such a way that they appear to have the same causal variant. And if that is the case, then you have sort of that forms a hypothesis that potentially this QTL, this regulatory effect, could be underlying your GWAS signal. And using this approach, we find thousands and thousands of GWAS loci where we find these kinds of indications. And in the latter part of my talk, I'll talk a little bit about how we can further probe at these questions with different types of approaches. But I'll mention here that this type of analysis of putative regulatory drivers of GWAS loci can be further enhanced by the added resolution that cell type interactions provide into these analyses, being able to pull apart signals that derive from different cell types. But to kind of like very briefly summarize where we are and what happens next, kind of after post GTEx world for us, thinking about functional genetic variants. Us and many others over the years have shown that regulatory variation is extremely common in human populations. It provides a lot of insights about regulatory mechanisms, its tissues and cell type specificity. There is still a lot of work to do in the GWAS interpretation, but this is one of those kinds of glass half empty versus half full type of a question where there are certainly some critics of the QTL approach, but there is no approach that is going to be a silver bullet, and then we have certainly come very, very far with these types of resources and analysis. And altogether, I think we can basically say that the cis eQTL or splicing QTL catalog across human tissues in Europeans is pretty much complete. We don't have every single tissue, but we actually do cover a lot of the ground here. And if you sort of like you could dig deeper, but this is a very, very comprehensive research right now. But there are still many further questions that we need to analyze using different approaches. So I've highlighted the importance of understanding cell type specific or cell type sort of driven regulatory associations, adding that resolution into these data sets of regulatory variants. Many people are pursuing that. The cis QTL mapping and kind of like these analyses, they are still underpowered for many analyses, including some of the more complex, let's say, environmental interactions, cell type interactions, and trans QTL mapping for sort of downstream pathway type of regulatory associations. These GTEx and many other analyses are really strongly strongly kind of focused on RNA sequencing as the molecular phenotype or the source of molecular phenotype kind of quantifications. But there are many other molecular phenotypes of great interest as well. For example, ETHZ provides really exciting, interesting views into chromatin activity and genetic variants on that, etc., etc. We have been doing some recent work here that basically probes into how to do some of these analyses with better transcriptomes. So basically using long-read transcriptome sequencing to analyze genetic regulatory effects and kind of more sophisticated resolution into the transcriptome space. GTEx is an American sample. It's mostly European ancestry, about 85%. And even though it allows some analysis of non-European regulatory sort of variants, now that there is a strong push to expand GWAS resources and catalogs and studies to the non-European populations that have thus far been underrepresented, it's very important that these functional genomics data sets are also kind of created in non-European populations to a sufficient extent to allow functional interpretation of those associations that are discovered. There are many kind of like quick walls that when people find non-European, for example, African-specific GWAS signals, and then there's no QTL data to look into potential functional effects. And then this kind of QTL mapping approach that GTEx and many other studies use, it has been like about 10 years ago or something when I started doing these types of work, it was the only scalable genome-wide approach to ask about molecular effects of genetic variants. But this is no longer the case with CRISPR and many other experimental tools that we now have, providing a very, very diverse and interesting toolkit to use completely orthogonal methods to ask how genetic variants affect gene expression and gene regulation and cellular functions. And we're basically pursuing all of these approaches in the lab. And now I want to spend the latter part of my talk talking a little bit about some novel experimental approaches that we have taken to basically follow up GWAS associations. So this work is a collaboration with Neville's and Jana's lab, another PI at the New York Genome Center, and spearheaded by our joint postdoc, John Morris. So what we wanted to do in this work was to kind of take a bunch of previously existing sort of bits and pieces of tools and combine them to kind of a toolkit that we call altogether StingSeq, where we use experimental CRISPR-based perturbations to characterize gene expression effects of GWAS loci. So how StingSeq works is that we first do fine mapping of GWAS signals to find putative causal variants. We intersect those with regulatory elements that we get from ENCODE and such databases to basically identify putative cis-regulatory elements, candidate cis-regulatory elements. And then we design CRISPR-I gRNAs, so CRISPR inhibition gRNAs, to target those loci. And we can then do those CRISPR inhibition assays to silence those putative enhancers in a highly pooled, highly multiplexed manner. And then use multimodal single cell capture in a single cell to quantify the gRNAs that they contain, the transcriptome-wide gene expression profile, and then also surface proteins. I'm not going to talk about the protein space today, but what we get from the gRNA and single cell transcriptome data is the ability to test in a pairwise manner for each gRNA what is the expression of basically differential expression, comparing cells that have a given gRNA and then cells that don't have that gRNA. And this is sort of like a very highly sort of scalable pooled CRISPR approach for probing at these putative GWAS loci in the non-coding genome. So the way this kind of approach works, just using one locus as an example, is that here we have a CHUV association, sort of LD patterns, a big peak. There is some fine mapping of a limited set of SNPs that are according to fine map approach, the likely causal variance in this locus. And then designing gRNAs to target these different putative causal variants. Here on the left, I show the results for the one that is shown here in red, where we can then analyze which gene's expression is affected by silencing the locus of this particular variant. And we can see here on the left, so here in these maps, each kind of like small gray dot is a cell. And we can see that in most of the genes in this region, the gene expression level is exactly the same in the cells that have that gRNA targeting this locus or not. But for CD52, we have a clear decrease in the expression of this gene when this particular enhancer is silent. So this is suggesting that CD52 is the likely GWAS target gene of this particular GWAS locus. And it is, in this case, it's not the closest gene. So when we do this analysis for a bunch of different variants, looking at 88 candidate cis-regulatory variants, we find that 37 of them actually have a significant gene in cis, so 42% of all the loci that we tested, which I think is a very high sensitivity. I actually did not expect this to work this well. The data looks extremely clean as well, so that if we can test the p-values kind of the actual targets, so looking at enhancers and nearby genes, we see a strong enrichment over the negative controls. And we can analyze the enrichment of these discoveries in the different types of enhancers implicated by different chromatin marks. And something that is kind of consistent with previous studies is that in most cases the gene that we find is the closest gene to the SNP, but this is not always the case. There is like a solid 30% or so of situations where it's not the closest gene, which is not something to kind of like be ignored. We cannot just assume that the closest gene is the one, because it's most often the case. And we typically find just a single gene for a given SNP locus or a given enhancer, but sometimes we find several. And we also have situations where a single gene is affected by multiple independent cis loci, multiple independent CREs. But one of the most exciting results for me from this study was the fact that in a couple of cases, we were actually able to find not just the target genes in cis, but also their trans effects. So an example of this and a locus that we characterize more deeply in the paper is a locus of the GFI1b transcription factor, where we actually have two independent GWAS signals, so LD independent signals, one of them in the GFI1b intron and one in a downstream locus that is, sort of overlaps some enhancer marks. And when we analyze the cis signal of these two SNP loci, we can see that both of them affect the expression of GFI1b to a slightly different extent, but basically two independent GWAS variants, both targeting this transcription factor gene. And then when we turn our differential expression analysis to the entire genome and ask for these two variants, and here I show also a third variant that I'm not going to talk about more, we see that they actually have an effect on gene expression to a large number of genes across the genome. And here, just taking the strongest GFI1b SNP, which is the downstream enhancer, we see that there is hundreds of genes that are significantly and quite strongly affected by the silencing of this particular enhancer. And when it comes to the sort of like what's the potential sort of importance of this pathway, sorry, I actually forgot to mention in the very beginning is that the GWAS data that we are analyzing here are blood cell traits, and we're analyzing these in the K562 sort of erythroid type of precursor cell line. So these two SNPs that in the GFI1b locus are associated to kind of like a diverse set of blood cell traits, a little bit of this and that, platelets, things, some white blood cells. But what is exciting for me and something that I was sort of extremely sort of positively surprised about is that when we looked at those trans genes that I showed you on the previous slides and the GWAS signal there, we actually saw that these trans target genes are also enriched for genetic associations for blood cell traits in the GWAS data, which is really suggesting that these blood cell traits are affected by general polygenic dysregulation of this pathway that is not just sort of like that these GWAS signals are not kind of like randomly scattered across the genome, but there's actually an enrichment of these in specific pathways. And here, particularly for platelet and red blood cell traits have a highly significant enrichment here. And we can actually dig a little bit deeper when it comes to these target genes. So taking these several hundred genes that show a differential expression after silencing of this enhancer with this GWAS locus, we can then do a co-expression analysis to look at how these genes may be arranged in a co-expression network. And we see 3 different clusters here, where the cluster that we just call A, that has this sort of like a very tight cluster, this has the highest enrichment of GFI1b ChIP-seq peaks, and this seems to represent the direct targets of GFI1b. Versus the cluster C being quite interesting, it's these genes are enriched in terms of heme biosynthesis. It's looking at kind of what cell types are expressing these genes. It's red blood cells and megakaryocyte progenitors. And when we go back to the GWAS enrichment analysis and do this by these different network clusters, we see that the overall GWAS enrichment that I showed previously for red blood cells and platelets is largely driven by this cluster. So we really think that this is kind of like one of the key clusters that sort of like actually ends up affecting sort of cellular differentiation and functions related to red blood cells and platelets. So to summarize, what we have here in this paper is an approach to discover GWAS target genes with an experimental approach that has a very high scalability and high sensitivity, a pretty high success rate in terms of finding these, especially cis target genes. And this is very orthogonal data to EQTL mapping and co-localization. A handful of the loci that we discover with STINGSeq are also implicated by EQTL analysis and co-localization. Most of them are not. And this is something that we plan to dig a little bit deeper into to understand sort of like, what does EQTL analysis capture? What does this type of analysis capture? Why aren't they in more of an agreement? But I think it's actually great that they provide orthogonal data, because that allows us to get a more comprehensive view. And I think it's interesting that while there is often a single target gene that is closest to the SNP, there are important exceptions to this of more complex regulatory architecture in these loci. And then these trans signals are highly informative in terms of developing hypotheses of the actual cellular mechanisms. But it's also, I think, noteworthy that in only 2 loci out of about 40, 50, we find these trans signals. So many of these GWAS signals, even though you have a clear cis effect on a given gene, there is no sort of following trans signal of more general regulatory perturbation. And some of the future challenges that I think we need to tackle as a field to be able to use these types of approaches to their fullest potential is to improve fine mapping, which is very important for functional follow-up. We here targeted very well fine mapped loci, and if you have just terrible fine mapping, it's difficult to do anything. And here, for example, more diverse GWAS data sets will help. We were, as I said, we're interested in the better disentangling of the EQTL signal or lack of it. And here we were able to analyze blood cell traits with a pretty good cell line that kind of works well, is easy to grow, easy to manipulate. But for many different diseases and many GWASs, these good cellular models that are sort of experimentally feasible and scalable are just simply lacking. And also, I think that in general, the better characterization of downstream effects is something that is going to be very important for getting kind of beyond the cis target gene to really kind of dig deep into this with the kind of trans target analysis and other approaches for cellular phenotyping. And one of the kind of communities trying to pursue these types of approaches and many other approaches to tackle the challenge of complex disease interpretation is the International Common Disease Alliance. So just as a quick mention of something that you may have heard of, this is a big international initiative to bring together people doing the sort of GWAS mapping type of things, functional analysis, medical applications, and then the different sort of data infrastructure, innovation, training, global equity components of the scientific enterprise to really push forward analysis of complex disease genetics. So this is something to keep an eye on in the future. ICDA is now sort of organizing itself to get in full speed. And with that, I'd like to thank a lot of people in my lab. John Morris led the STING-seq work, and a lot of people have contributed to the GWAS work. Very special thanks to Francois Aguet from the Prod on that, and thanks to collaborators and funders, and conflicts of interest are there as well. Thanks.
Thank you, Tuuli. Thanks for this nice presentation. So we have time for a few questions. I mean, the advantage of being in person, we could go and raise questions. So feel free to ask a question now, if among the board or among the panelists, or through the Q&A again. Questions? Do we have any? Let me shoot one then. So one of the aspect which is we are basing everything onto a reference genome. So how much do you think we're still confounded with the CNVs and structural variants when we call QTLs and EQTN? And how much do you think the QTLs are still troubled by that?
Yeah, so
about the telomere to telomere paper that just came out a few weeks ago with 200 megabase more genome.
Yeah. So we actually back in the days when I was in Manolis's lab, we published a paper like sort of showing that alignment biases are not drivers of kind of false positive signals in EQTLs. But that was a while back, and it would actually probably be good to revisit that question now that we actually have better quality gene. Everything is just better and bigger. I don't think that like CNVs and structural variants will have such a big effect. So if they are in the non-coding space and they're kind of like the causal variants, they will be either captured by, like here we used whole genome sequencing data, or attacked by SNPs, just like any variants. And it's not that common to have like entire genes or big parts or subsets of genes deleted. That's somewhat rare because of, I mean, that would have often pretty terrible functional consequences. So yeah, certainly something there, but I don't think it's going to be a major thing.
So there is a question from Zoltan. In the last result you presented, would the lack of trans effect of fine-mapped GWAS SNP contradicts the core peripheral gene models proposed by the Pritchard group?
Yeah, very good question. I don't think it contradicts, but it raises the question, or kind of like, I think, and they published an interesting paper that is somewhat kind of using related experimental approaches to kind of probe at this question. But I think it's an interesting question. I mean, they basically state that a lot of these GWAS, these small GWAS signals would be kind of regulatory effects of these kind of core genes that then affect GWAS traits directly. But if we find a lot of these kind of GWAS signals that don't have regulatory effects, at least as far as we can see, if they kind of affect the GWAS trait directly without a regulatory mediator, that would indeed be against their view. But it's of course like, I mean, we analyze one cell line, and who knows what kind of effects these things might have in some other context, and kind of, I think it's complicated. It's kind of, it doesn't support the omnigenic hypothesis. It certainly does not falsify it either.
So the jury is still out. Thank you very much, Tuuli. There seems to be no more questions. the next speaker that arrived at 2 o'clock in the morning because the flight was delayed, as far as my spy tell me, is Nicolas Katsanis. Sorry about this. We tend to call people differently in Greek. He's a human geneticist and cell biologist that led several groups, led groups in Baylor, Johns Hopkins, Duke, Northwestern. I hope I didn't forget any. And he has a love for head size of fish. That's the first time I met him, and seems to be adamant about the size of the head. As quite a lot of Greek scientists lately, they tend to go to industry or either start companies and try to deliver new opportunities. And he's now at Rescindo Therapeutics, where they try to rethink the drug discovery for rare disease genetics. We were in the middle of a pandemic, so when companies are sharing news, it's kind of hard to be on the headline. But what is quite amazing is that Rescindo actually started the phase 1 clinical trials that got approval from the FDA in September 2018. And in February 2021, they received the FDA approval for orphan drug designation in rare pediatric disease with a compound called RRC57 for the treatment of Kabuki syndrome. And this is actually something that I think is perhaps a trend that some of the basic scientists needs to transition and bring their science in the industry and try to make a change and transform the field. And Nikos, without any further ado, we are very happy to have you here. And I hope the time you slept was sufficient to give this talk.
Thank you. Hello, everybody. I'm so sad that we cannot be together in person. I was very much, I'm sick and tired of carrying on professional and friend interactions and friendships through Zoom, but hopefully this is the last one. So thank you for the introduction. I'll see if I can be really brief. Yes, I'm one of those who left the nest to the private sector, but what I do actually is because life is too boring to just run one company, so I run two. And I think it's going to be important because the world we live in will have to figure out how to combine analysis of big data as well as with experimental studies. So I'm actually, like Ioannis said, the CSO of Rescindo Therapeutics. This is a company that's been around for almost 6 years now. And it is true, we're currently now in clinical trials with one asset, and we're very close to clinical trials for a second. Exciting times. But more recently, I actually co-founded another effort, Galatea Bio, and this is a data analytics company that's looking to harness genomic diversity as a method for and modularize the human genome as a method to get to new drugs. So what I thought I'll talk about the BG time before genome. So back in the day, and this is when I was also a very young pup and I didn't know what I was doing at all. Sort of drug discovery had they did have a sort of medieval flavor to it. Basically, it was throw spaghetti on the wall and see what sticks. Many people, they were looking at phenotypic screens on core cell phenotypes. You had usually some cancer cell line. It had some sort of behavior, or it fluoresced, or it luminesced. You threw some ungodly number of compounds on it and said, these ones worked. Eye of Newton, wing of bats, as far as I'm concerned, and you try to guess which ones were the better candidates. And then, of course, you did an animal study. But they used to be the mouse for no particular reason, other than it was a well-established experimental animal. And then, the failure rates for transitioning from preclinical to clinical studies were exceeding 99%. But then you had to go back and do over and over again. And this was one of the factors that has actually led to very high R&D costs for drugs. And I actually think for those of us who are in drug discovery, we need to be thinking about how much things cost, not just what they do, because what is the point in developing drugs if only about 10 people on the planet can get them, and the rest of them will become bankrupt, 5 or 6 years later. Now, of course, the world has changed, not only in genetics and genomics. I was very glad to be part of the train and to watch and play a small role in the evolution of genomics, genetics, and medical genetics there in the past years. But if we look at where we are now, there's many changes in drug discovery, and I'm just going to highlight 4. The first one is now we're getting ever larger cohorts with associated either electronic medical records or prescription data. We're getting better and better at things like natural language processing. We'll be getting better and better at standardizing phenotypic data using HPO terms and others. I think this is going to be important because we will need to develop a new set of language to share the information. And I think we are moving into a world where we're beginning to federate clinical data, and we're breaking down the whole sort of fiat model of, University X is holding all the data for this and clinic Y for the other. And I think that's exactly right. And I applaud these efforts in the same way that international consortia came together to solve the problem of genomics at scale. I think the same thing has to happen with sort of genophenome, as it were. The other thing that sort of, might have flown a little bit under the data for some people is that there's been incredible progress in solving protein structures. At the moment, we have about 45,000 structures deposited in public databases. And now, given that we have much better computational tools, machine learning, things like that, not only are we able to solve computational protein structures at some reasonable degree of resolution, we're also beginning to start to see how they behave in space, how they bend, how they flap. These are very important things. The other thing that's going on, it's still imperfect, but we're getting massive and more accurate PPI networks. And of course, machine learning is becoming a significant component simply because the size of these data sets cannot be used in any other way. All right. Among all the beautiful things that we, the scientists occupying this planet, have done, and all the fancy tools and AI this and all that, the fact remains that if you at the very least look at post hoc of what worked, you go back to your work and say, okay, we've tried 100 different things. Human genetics remains among the strongest predictors of success. And when I talk about success, I really talk about clinical trial success of a new asset in people. This is important because we have to get the success rates higher, both to expedite drug discovery, but to also solve the economics problem that drug discovery faces at the moment. All right. So then the question becomes, can we leverage sort of smart genetics to improve biology and drug discovery? And there's many answers to this, and I don't have all the answers, but what I can do is show you sort of two pieces of work that took a fairly short period of time to get us there. And I don't know how scalable they are, but there's certainly sort of two ways to go about things. All right. So I'm just going to share two examples because these are the two examples that are moving pell-mell into the clinic right now. The first one is from functional screen to new leads, and the second one is so from serendipity from gene discovery to new treatments. Okay, so the first one, I've been using this slide for years. I absolutely love this idea. And the fundamental thesis of this vignette is that not all deleterious mutations are deleterious. I have always sort of reminded myself that when we talk about pathogenicity, we're really talking about two different levels: pathogenicity at the level of protein function and pathogenicity at the level of phenotype. And many times one is linked to the other, but this is actually obviously not ubiquitously true. So we started thinking about how you can use genetic suppressors as targets for drug discovery. There's many ways to identify suppressors. These days, folks are beginning to harvest very large genome data sets for resilience alleles, and I think that's great. In this particular case, we use a functional approach. So this is a story about the ciliopathies, a group of disorders that some of you might know is very near and dear to my heart. I still dream of the way that I'd be able to call up all the moms and dads of children with ciliopathies that I've interacted with the last 20, 25 years and tell them we're finally there, enroll in the trial, we have a really reasonable lead. It is a fascinating group of disorders, in part because of its pleiotropy. I'm not going to talk about the genetics of these diseases. The work has been published many times over. But I remain fascinated by the fact that we have a group of disorders that are progressive, but also structural, developmental, and adult. And we know that part of the reason is because of the near ubiquity of the organelle, the primary cilium. We now know that this organelle is present in almost all cells of the human body. In fact, it's the exceptions that are interesting, like T cells and hepatocytes. But it has been a very difficult nut to crack, because it is first of all, if you have a structural defect in an organelle, how on earth do you go about restoring the function? And two, what kind of molecule would you get? And of course, there's delivery issues. So the way we approach the problem is we said, forget what we know, because if we go after just what we know, then all we're doing is searching under the proverbial lamp. And the one thing I've learned over the years is that intrinsic bias tends to be the greatest impediment to progress, because we all become so deeply enamored with our own ideas, not because we're vain, but because we're human. So we keep following our own ideas, and we develop blind spots. So what we did back in the day is we said, well, how can we design a rational paradigm? And the idea is to go sort of reasonably agnostic. And the only hypothesis being is, can we identify genes and alleles that might rescue a credible ciliopathy phenotype? And because these disorders are very rare, and because it is going to be very hard to imagine a world in which we will develop meaningful statistical tools to actually identify these sites, because most of these are two-way, three-way, four-way gene interactions, which means most studies will be underpowered. We actually did we took a rational approach. So sort of old observations, but as it turns out, these are the Bardet-Biedl syndrome proteins right here, which are things that we were involved in cloning and characterizing for many years. So what was surprising was that a number of years ago, we reported that several of the BBS proteins actually seem to be regulating the function of the proteasome. And this was a little bit shocking, and at the time it was a little bit controversial. Of course, since has been replicated a number of times. It was shocking because this is not what the expectation was. But it turned out to be true, and it turned out that one of the functions of the primary cilium and the machinery rather underneath the primary cilium is to regulate the degradation of effector molecules, paracrine signals. So the idea is that you receive the signal. In this particular case, I'm showing you Wnt. Sony Hedgehog is another famous example. Notch and F-kappa beta can go on. And then what happens, depending on what you read up there, you have some decisions to make. And in this particular diagram, the decision that you have to make is whether you're going to degrade beta-catenin. If you don't degrade beta-catenin, you're going to have more of it in the nucleus. When it goes to the nucleus, it will bind to TCF-left elements, and those will drive transcription of a number of genes. So what we were able to show many years ago, at the time when we did this experiment, we did not understand it, not truly. This is a very simple luciferase assay activity, where what you have is you have a series of concatenated TCF-left elements. And then when you have activated beta-catenin, you have increased expression. And what happened was that when we at the day suppressed BBS1, but also BBS4, BBS6, we started seeing increased expression. And of course, I'm not going to show you the data of a 14-year-old paper, all the data, but it turned out that this is true because this step here is failing. We're not degrading beta-catenin by the proteasome in the cytoplasm very well. So on the base of that, we made a system where we made these concatenated TCF luciferase assays, and we generate a conditional knockout of BBS4. And it had to be conditional because we need to control the experiment. The cells that are missing ciliary and ciliopathy proteins behave differently when it comes to responding to cell cycle cues and things of that nature. And then we laid these cells on top of arrays that had siRNAs for 22,000 genes. These were pooled RNAs. This was 134 plates. Each gene, the genome, was represented in 2 different pools by 5 different siRNAs. 72 hours after plating, we stimulated the cells with Wnt3a, and of course at this point 0, we also took out BBS4. 72 hours we stimulated with Wnt3a, and then a little while we harvested, tested them for viability, and look at the data. Okay, what does the result look like? So when we applied a very stringent filter of Z of -3, and of course significant p-value, we end up with 29 genes. What does that mean? That means we found that there were 29 genes in the genome that, when suppressed, could rescue the aberrant luciferase activity induced by BBS4. When we did a second experiment, AXIN2 is a very well-known target of beta-catenin, so just qPCR, 13 of the 29 genes survived. And this is kind of important because it is not very common that you get about 40% harvesting of the secondary screen. And that told us that we're being quite stringent here, which is a good thing, but it also told us there's probably more hits lurking underneath our threshold, because we actually mandated this to be present in both replicate sites of the primary screen. And then we did, genome harvesting, which is things like look at tolerance for nonsense mutation, association with disease, because as everybody can imagine, of course, there's not much value. I should say, there's enormous intellectual value in understanding how these 13 genes, the suppression of them, can actually rescue a ciliopathy phenotype. But most of these guys, well, more than half of these guys, are either themselves implicated in a different disease, or they are clearly intolerant to loss of function variation in the genome, or there's an animal model, a mouse, a rat, a worm, a fish, that is showing such severe pathology that the idea of attacking these things as a drug target is not going to work, because we're going to have such adverse side effects in humans that it doesn't work. Okay. So thinking about treatment modality. So, we can all do these screens and we can say this, that, and the other, but the fact remains, does it work? And we tested this modality in zebrafish embryos. I remember when we first started this saying, zebrafish, you test everything in the mouse. Of course, now we know that the zebrafish model is accepted by the Food and Drug Administration as one of the preclinical models. We have now seen, I think, the first molecule gain FDA approval that originated from a zebrafish screen is going to go more and more. It's just a question of sort of getting to new habits. There's no magic involved. What you are looking at here is cross sections of the photoreceptor of a BBS4 mutant in this particular case. We've done morphins, we've done mutants, we've done crispins. I'm sorry, this is a crispin here. And what we're measuring is the abundance and the position of rhodopsin. And the reason we're doing this is because in most retinal degenerations and in all the ciliopathies, rhodopsin gets trapped in the inner segments. The idea was that trafficking of rhodopsin was not working well between the inner and the outer segment. However, we now also think that a contributor to the amassment of rhodopsin in the inner segment is also the fact that rhodopsin is not being degraded very well. We know that accumulating rhodopsin is toxic to the photoreceptors, and we know this because when colleagues generated a mouse transgene where expressed wild-type rhodopsin in the inner segment at high levels, these animals also develop retinal degeneration with a slope very similar to what we see in humans. And what you're observing here is that you invert this image and you map the quadrants. In the beginning, we used to do this by hand. Of course, now everything is being done by image recognition, because you can write pretty reasonable algos to do this. And this is what the data look like. So I should tell you here that the key lead compound, the key lead gene that came out was something that was called USP38, and it was a deubiquitinase, which is a lovely, lovely result, because we had a proteasomal hypothesis which we chased, but then from a genome-wide study, a ubiquitinase fell out. In fact, 4 ubiquitinases fell out, but this was the one that had the best metrics and the best profile for humans. And what we found was that this is the phenotype, this is the fluorescence of rhodopsin, and as you can see, when you actually execute a double mutant for BBS4 and USP38, you restore to wild type. This is also important. This is a functional test of photoreceptor, where what you actually do is you shine a light on the fish. You induce light-dark transitions. When animals are exposed to light-dark transitions, they change their activity. And but if they cannot see, of course, they cannot change their activity. And once again, we have an improvement there. Okay, that's the good thing. And I got to say, everybody gives these talks about, look how great we are. Well, look how great we are. This was years of work. Now, the bad news. we were very, very, very proud of our accomplishment. We were going pell-mell. There's actually a pair of paralogs, USB 35 and USB 38. And then we go to our medicinal chemist friends and saying, what do you think about this? So we do this by CRISPR, da, 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 da. And they shook their heads and said, this is a terrible target for drug discovery. And it's a terrible target for small molecule discovery because one, it's a reasonably globular molecule, so it's very difficult to find a place to attach something. But most importantly, it is very similar. They're evolutionarily derivative from USB7. And if you kill USB7 in humans, very, very bad things happen to you. So it was, I remember the day that sort of we got this news, and it was a very bitter pill to swallow because, you know, your dreams of drug discovery is gone in that regard. And, you know, several years of work are down the drain. Sure, we wrote a nice paper and all this, but whatever. You know, there's been 2 million papers written since 2000, so 2 million and one. But so these were the options ahead of us. And it was a pretty depressing time. And I think, you know, we all spend our time talking about our successes, but we also should be spending a little bit more time talking about our failures. And it was a depressing time. It was a demoralized time. We didn't know what to do. And then David Brun, the company, actually came up with a really decent idea. He said, hang on. He said, sure, we know that this gene that we pulled out from the genetic suppressor screen is not going to be a good drug target. But we've also learned that the process is actually targetable. So what if instead of targeting the gene, we actually target the activity of the gene? So in other words, actually find a molecule that can actually mimic the suppression of the gene ab initio through a different pathway. So it's like, okay, David, you know, this is where you sort of take a deep breath, you spend some money, and you hope for the best. So here we took advantage of we engineered a cell line in which green fluorescent protein is fused to unstable GFP. So GFP has received a degron signal that tells the cell to degrade the protein, and the degradation actually happens through fast ubiquitination. I don't want to go into the mechanics of this, but and the question was very simple. These are cells, docile in silence. If your cells are sick, if the proteasomes are not functioning very well, then you're going to get green cells. And once you get green cells, then this becomes reasonable for a high content screen. We tested this as a proof of concept with a known compound that can enhance the proteasome function, this MBC. And just in case everybody gets excited, this therapeutic, this is one of the antifungal, antivirals that people put on the contact lenses. And if you were to drink it, it will burn your trachea and maybe kill you afterwards. So maybe not a good drug, but nonetheless, we saw this very steep drop. Actually, this is not a good drug curve drop, but nonetheless, it's function. And we actually perform a focus screen. We started with 5,000 compounds, and these are all FDA, EMA, Japanese Medicines Agency approved compounds. For many of them, we have toxicity. We had the sensor. And the key to our success in that particular case, because yes, it did work out. See, I already told you the end of the book, is that we have a credible, quantifiable phenotype in an animal system to measure the pathology. So we identified 2 strong leads and 4 backups. Both of them are known, approved FDA drug. In fact, one of them is a generic, which creates commercial problems, but that's not for this talk. And this is the key experiment with our 2 compounds, C11 and C27. So once again, I'm showing you the startle assay, which is you acclimate the fish, and then you have these cycles of dark and light transition. These are done on an instrument where the camera records the movement and the changes, so you can avoid operator bias. And this is the picture that you saw before with the photoreceptor slices. Only in this particular case, it's not a double mutant, but these are animals that are bathed in drug from 1 day post-fertilization, and they're measured at day 5, and the results are gorgeous. We were so happy. And once again, if you look at the number of responses, you get between 3 and 4 responses of the fish in the exposure to the drug. They go blind, they can't see, so they barely respond. This is fully restored by both. And likewise, the fluorescence is fully restored. And the thing here is that both of these drugs have very, very high tolerance, are very well tolerated ingestibles in humans that have been used in many applications. One of them is actually a very well-known antiviral. So we're excited. We're in the middle of preclinical, of completing the preclinical studies right now. We're headed to the FDA by the end of the summer, hopefully with some good news. The second thing I want to talk to you about today is this notion of surprising gene discovery, and to remind myself more so than anybody else that agnostic discovery does have absolutely intrinsic value. And the agnostic discovery is I still remember the day that Bernd Volnick contacted me, and he said that he had a kiddo who walked in his clinic with Kabuki syndrome. He had performed whole exome sequencing, and it had turned out the only thing that had turned up was not mutations in KDM6A, came through the methyltransferases. It was a mutation in a gene called RAP1A, and the kiddo had essentially converted this through paternal isodisomy. Uniparental isodisomy of the paternal chromosome had turned this a chunk of chromosome 1 into homozygosity. This was one of the few predicted deleterious rare alleles in that chunk. Of course, we tested the zebrafish model. Of course, it turned out to be pathogenic. We published this paper many years ago. What was surprising was that for Kabuki, everybody has been thinking about regulation of gene expression, and of course they should. It's methyltransferases. However, the problem with the methyltransferase is that you can't understand the specificity of the phenotype. You can come up with hypotheses, but it is difficult to understand how KDM6A or KPTM2D mutations, potent drivers for cancer as well, will give rise to sort of diversity but high specificity phenotype. Well, it turns out that RAP1A has been studied before, and depending when you look at, and this is important, steady state versus development, it actually acts as an activator of phospho-BRAF or a repressor of phospho-RAF1. So we actually tested to see which arm of which flavor of the RAS signaling pathway was true in a developmental model of Kabuki syndrome. And the key experiment, this is an old experiment now, we're looking at the downstream activity of BRAF, which is phosphorylation of MEK1/2, and we see relatively increased phosphorylation there, arguing that we're in the right pathway. But this, to me, was the key experiment. This was early convergence and extension, and this was length of the jaw. Again, we published this paper. I don't want to go into the data too much, but the important thing to say is that when we co-suppressed back in the day RAP1 with RAF1 together, we actually saw rescue of both of these phenotypes, telling us we're probably in the right ballpark. So the good news with RAS inhibitors is that they have been studied to such a great extent for neoplasias. And also we were very fortunate in that we were in an environment we were able to grab in the pre-competitive space, a lot of these. So we started with 27 compounds. Some of them are 2 compounds and some of them were close relatives of clinical compounds. Unfortunately, regulations prevent you from testing some non-clinical compounds because companies don't want to expose themselves to risk. That's a different conversation. But the key thing was we actually found a BRAF inhibitor that rescued phenotype very, very well. A dead version of the BRAF inhibitor where it was a small tweak that it cannot dock to the kinase domain did not have an effect. And this is early development, and this is the jaw. Children with Kabuki syndrome have a very short jaw, the back of nothing. And of course, anybody who looks at this data goes, because convergent extension is an early gastrulation movement. The jaw, who cares how long the jaw is? These are not pathways relevant to children's health. However, this is. And this is now generation of new cells in the brain. Now you're looking down at the fish. In fact, the retinas are right here. And I like this assay better also because it's quantitative. And what we're able to find is that we were able to impact neurogenesis. And what we're able to show is that not only for RAP1A, but both for KMT2D and for KDM6A, In fact, we've now tested all the known genes for Kabuki syndrome, and my statement holds true. For every gene that we've tested for Kabuki syndrome, we can improve neurogenesis by exposing the animals to the BRAF inhibitor. The next question, though, was, well, this is very early in development, which in humans would translate in utero. Can we actually, is this meaningful? Because in children, we would have to do this postnatally. And I'm just going to show you the very, very brief summary of the results. This was done in Bill Wetzel's lab at Duke. This is the what, when, where test, where you have fun, where you take objects and also novel object recognition. These are tests, these are cognitive tests primarily for learning and memory. And essentially what you do is you allow animals to equilibrate, you offer them a set of objects, A and B, and then you test and then you move them around and you ask the question whether the animals actually have exploratory behavior, what they do. And again, I'm not going to show all the data. Here's the important results. We injected intraperitoneally the BRAF inhibitor in animals that were 14 days old, and we did this for a week, from day 14 to day 21. We also did a 2-week paradigm, and the data looked exactly the same. This is important because children with this BRAF inhibitor has been tested for pediatric glioma, and they seem to be tolerating the drug for administration continuously for at least 6 months. So in our paradigm, we did a 1-week testing, we also do a 2-week testing, followed by a 2-week holiday where you do not give drug to the mice, and then we assess them at P30 and P40, and we observe complete rescue of the neurogenesis phenotype with this particular BRAF inhibitor. What is important is when we used a MEK inhibitor and an ERK inhibitor, we did not see this rescue. We do not understand the specificity of the rescue, but there you have it. And the other thing to say is that the amount we used was only 50% of the human dose, 5 mg per kilogram, which means that we're well within the tolerated range. As Ioannis said, we have obtained FDA approval. We're actually now collecting sites with the middle clinical trials, and we hope to have good results soon. And at the end of the day, you know, what do we do this? I remember I was there. I was finishing up my postdoc when the human genome papers came out with great fanfare and all these things. And at the end of the day, we did all this, yes, to improve human knowledge, yes, to understand the body, but also to make her feel better. And for those of us who've been in the war zone for a long time and have chosen to transition to the private sector, is not fame and glory, is really to make it feel better so I can sleep a little bit better. So I'll stop there and take any questions that you have. Thank you.
Thank you, Nikos. So questions for Nikos on his research that involved fish. Have to.
I mean, you know, it cost $1, between $1 and $1.20, depending where you are, to house a mouse. It costs about 5 cents to house a fish. If you need to power a study with 10,000 individuals, would you do this in the fish or would you do in the mouse? You just need to know what the limitations are, and once you figure that out, then that's fine. So we don't study lung fibrosis in fish. No.
Of course. But the good thing is the FDA actually at one stage sees a light, and this is time.
This was a watershed. This was a watershed moment, and this was because the OECD put together a panel of individuals to evaluate the utility of this model organism. They started working in 2008. I want to say I might have my dates wrong, and they came up with a recommendation in 2014, and by 2016, the FDA issued this new directive. Again, you know, it's the usual. You need the right controls, you need the right numbers. It's like everything else, but fish.
Fish, beautiful model.
It's a beautiful organism when it works.
When it
doesn't work, it's a piece of crap.
Questions? Otherwise, I'm going to talk about fish because I love genome duplications. Manoli.
I can ask a question. Hi, Nico.
I feel like we haven't spoken in ages.
What?
Nothing. I said, I feel like we haven't spoken in ages.
Exactly. It's only a few hours. So I guess the question is, you made the point, and I think that's an important point about drug discovery informed by genetics.
Yes.
But it seems to me that there's sort of two subcategories of drug discovery driven by genetics. Those are driven by those kinds of things that you've been doing, which are more targeted on specific, I mean, it involves screening and so on, but it's more targeted, experimental-based follow-up of specific processes and pathways. And there's a more general one of let's just capture GWAS or do sort of mutation screens, find the PCSK9 equivalents, and then design, antibodies against it or this kind of thing. Which one do you think is more likely to give us quick results at scale? And by that, I mean a number of solutions rather than a highlight solution in the coming years.
Yeah, so this is a great question. And this is the one I've been thinking about very hard as well. Because obviously you can't be doing function, you can't go every indication, develop a cell line, So to me, whoever manages to merge PopGen level data with biological information wins. Here's the problem with the PopGen that I see at the moment. There's too few wins. So you look at BioView, when you do what you do, you come up with 5 targets from genetics. Well, it turns out 4 out of the 5 targets make for crap drug targets. Well, what have you left with? I feel that there's a lot of value left on the table because we are constrained by this sort of genome-wide approach where the whole genome is the denominator. So I have the sense that if we're able to combine biological observation to read in convert the genome into modules, I mean, or combine it different data sets, I think that's going to be the real win. We saw from Regeneron, the beta estradiol story that came out for glaucoma, and that's beautiful. I don't know if this is what is targetable or not, but again, this is one example. So that's one thing. The second thing is, and this is something that we're focusing on the Galatea Bio side, not so much on the Rescindo side, is improving the diversity of genome data. So I see incrementally short value in understanding more Northern Europeans and getting more Northern Europeans genomes and EHRs and all these things. And I see a lot more value in getting Samoans, Northern Indians, Mexicans from the East Coast to the West, and so on and so forth. So that's my answer. It's a little bit of a woolly answer, but that's where I see the world headed. Diversity of EHR data, and then the ability to functionalize modules and then test drugs. At the end of the day, we have to be able to not just derive the targets from PopGen, we have to be able to test the target. So we need to build scale for both of these things and combine the data. So are we willing to accept a set of hits that lights up as a protective allele, p-value of 10 to the minus 5 instead of 10 to the minus 8, if we have a biological result that also concords? Can we actually figure out how to harmonize these two types of observations? Very difficult. Can we defeat the old Fisher problem, right? Where it's either quantitative data or biological observation.
Yeah, and I think one of the issues is that many people have been sort of looking for the big effect size.
Yes.
They kind of consider the small effect size as irrelevant. What I think that's not true, because even small effect sizes, they just simply represent what's the current state of explanation of variance in the population. You could easily modulate a small effect size variant, let's say the gene where you have a small effect size variant, in a large way and get actually a result that is, proportional to your modification.
And that's exactly right. And the other thing to say, I mean, I think about retinal degeneration, for example, right? If I can reduce the rate of photoreceptor cell death by 10%, that's going to buy the patient years of light. So let's not knock it.
Marc, you're muted.
Nico, did I understand correctly from the Kabuki example that the general strategy for finding a treatment for a rare disease would to repurpose drugs that are already approved, FDA approved, etc. And because otherwise it end up with big hurdles after that, right? And
so it's I'm sorry. Yes, in this particular case, although you touched a very sore point. So I had conversations with a very well-known pharmaceutical company, and they were going on and on about first in class. And I was saying, but that doesn't make any sense, because a first in class automatically means $50 to $100 million and 7 to 10 years preclinical development, medchem, efficacy, this and that. Why? And they said, I'm sorry, I don't want to offend anybody, but the answer was from the Neanderthal days. They said, well, unless we make $1 billion, we're not interested. And I was like, it's not about the absolute number, it's about the delta. If you're going to spend $250,000 and make $1 billion, that's a 4x. How about if you spend $10 million and you make $200 million? To me, that's a better deal. Don't just look at the total number, look at the relatives. And people hum and haw. The fundamental issue is that we still have to solve is non-scientific. So the drug that we have is the composition of matter is owned by Novartis. We've been trying to get a license from Novartis to use the drug for Kabuki syndrome for 6 months. I'm going to publicly say they should be ashamed of themselves. It's a company with an $8.3 billion a year R&D budget, and here we are talking about some that will cost maybe $10,000. Their financial contribution is $10,000, and we're running all the board. It's like, well, this and risk and the other, and fundamentally people don't care. So that's the problem that we have to solve more acutely. So I'm a little bit bitter because here we are, we come to a potential solution to a devastated disease, no solutions, and we're stuck on 6 months is a long time, but we're stuck on legalese and we're stuck on indifference more than anything else. So this is the weakness of the repurposed drugs, because if you go with a repurposed drug, you're dependent on whoever owns the intellectual property to actually move the asset forward in the clinic. And by and large, the environment in the ownership of these things is not consistent with the speed of thought and movement that we need to see in genetics to get this thing forward. I do not know how to solve this problem. I do not know how to improve humans. So we CRISPR out bad attitude from business development types on?
Perhaps bring them back a bit of ethics, which is sadly something that is lacking. But ethics is a word that I think Kelly will probably touch upon in the panel discussion. If there is no more questions, and it's on record, so we will send that to the Novartis CEO so that we could change his mind.
No. I mean, we're making progress. It's just a matter of when you deal with large companies, it's like dealing with the Tower of Babel. You just got to get off to the right floor, talk to the right person, and get somebody who's
lots of management layers and lots of overhead until you get to talk to the right person.
That's right.
Without further ado, thanks very much, Nikos. Thanks for your talk. Thanks for all the speakers that gave this afternoon time. I'm sorry, we don't have a coffee break, but since we are on Zoom, we don't need a coffee, basically. So we can heads up directly into the panel discussion, and I leave the floor to Jacques to take on board this. Jacques?
Thanks very much, Ioannis. Hello, everyone, and please don't hesitate to grab a coffee. I mean, we cannot stop you from doing that. And I'm very happy to welcome the 4 panelists. So let's be frank, this has been world-class science talks presented today. I'm extremely happy. And we wanted to complete this day with a panel trying to come back to something more practical, which unites all of us. I think if we are here and if you're still around, that is because we believe genomics, human genomics can bring something to healthcare. And the question now is, what are the opportunities, short and maybe not so short term? And for that, thanks very much to the 4 panelists. So you heard already Kelly Ormond this morning, you heard Samuli Ripatti earlier this afternoon, and joining us now are Marc Abramowicz, Head of Medical Genetics at University Hospital Geneva, and Olivier Michielin, Head of Precision Oncology in CHUV Lausanne. So 2 frontline practitioners of the world of genetics, genomics we've been talking about. So this was planned for a full hour. We are half an hour behind, so maybe we'll stop after 45 minutes. Let's see how enthusiastic you all are. I invite the audience to use the Q&A feature of Zoom to ask questions, to put remarks that we can share between the panelists. And to start the discussion, I will maybe just ask each of you to pick one hurdle, to pick one reason why we are not yet doing genomic-based medicine for the most part, because we heard all the magic that could be, that will be one day, but today we are not doing it, at least in our hospitals here in Switzerland. So what is for you today the main reason why we're not doing it? And is there an easy solution to it, or is it unsolvable and will be in trouble for years to come? So Marc, I invite you to start if you agree.
Oh, with pleasure, and it's an honor. So I think we should distinguish what we already do and what we plan to do. We already do genomic medicine for monogenics. We have lots of evidence of success with monogenic disorders. The question is for multigenics, the polygenic risk scores. And I think that we heard some enthusing data today that we are really on the verge of doing it. And then I should pass the hot potato to Samuli to better speak about it. But the point is that if you look at disorders like, yes, CAD and breast cancer, why not do with polygenic risk scores what we've been doing for centuries in medicine? After diagnosis something very empirically, try to interfere, see what happens, and then move on, instead of trying to have long steps of translational studies be performed before we do that, okay? So for monogenics, we couldn't even consider that because there were so few patients. But with multigenics, well, some minds would tell you that you have to do that first. And I think we have very good reason to say that we should not do that first. We should do that in the course of empirically treating.
I really like that approach. Yeah. Okay, so maybe Samuli, you can take over right after that, because obviously you describe what is the cutting edge of PRS, and some people in your immediate surrounding are trying to do those clinical trials. This clinical utility proof that is lacking now. So how far are we from demonstrating the clinical utility and then having the argument to go to the healthcare insurances and to the politicians and tell them, look, that it's even cost efficient to do it? Do you believe we are getting there?
I think you mentioned cost efficiency, and maybe that's one important way to argue as well, that we can of course argue how many cases we would have, how many CAD cases we prevent, time windows, or that these type of calculations, how much euros are we saying if we prevent those? How many breast cancers would we see much earlier than what we are currently doing? How many breast cancer related deaths we may be preventing by intervening, or at least intervening early, either in terms of prevention or at least detecting the cancer early. So we need these types of evidence, I think, on top of what we already have, and I'm sure they are coming, and I know that many groups are working on providing those information. So that's one line of evidence. I think that's there's scattered information on those, but once we get our act together in providing a comprehensive overview, I think that's sort of done. Then there's the other perhaps more communication related, or we need to make sure that all the sort of the areas that are now doing the monogenic work and people like sort of share the enthusiasm that Marc just presented in terms of getting those talk our language as well. And maybe there's a little bit of language barrier now, but I think that's something that we can that's just getting to know each other better and get them to learn the way of thinking that sort of the more quantitative approaches provide. I think these are the two hurdles at least that are important, but definitely something that we can work on.
So Olivier, for you, the question is maybe slightly different because you are in a field where genomics is being used, but it could be much more, obviously. So what's, according to you, the hurdles now, or where should we put our energy to move forward?
Yes, absolutely. I think arguably in oncology, I think we are witnessing now this transition from a mono driver view of the disease to a more comprehensive view of all the alterations and what's the interplay between all these to resist to treatment. So I think we are already at the stage where it's perfectly justified to do small panels to understand the disease at baseline and at relapse. And so we're really moving towards genomics pretty fast. In oncology, as you mentioned, Jacques, we have a readout which is much faster. We'll know within a few months whether the patient is responding yes or no to an action which is based on the genomics data. So I think it's from that angle we have an advantage of having quick answers to our question. But we're also moving now towards even more advanced metrics, for example, tumor mutation burden that we need to measure, and that's something that requires a significant amount of sequencing to be able to call how deviating a tumor is from the healthy cell immunologically. So clearly, this is already happening in oncology, so to speak.
And Kelly, if I can rephrase for you the same question. So you saw the enthusiasm of the US and Silicon Valley, as you mentioned. You now discovered the Swiss carefulness or slowness, or even the Swiss-German problems with gene in general. There is some reluctance to use genetics for all kind of bad and good reasons. So what's your impression? What, from your perspective, is preventing us from really trying to implement genomic medicine today?
Yeah, we did have the same problem with primary care providers, even in the Silicon Valley, this notion that we have all this underlying knowledge and how can we make it the most relevant for those primary care providers who are with the patients every day and make it easy for them so that they don't have to master this whole new complicated set of knowledge. So I feel like maybe if we can distill this down to the core things, have some very clinically applicable tools for our care providers, and then help them with the support on the back end. As I was working with primary care providers in the Stanford clinics, they needed someone to help them say, okay, this patient, I understand the monogenic referrals, I've got that, but maybe this next patient, it's a little subtler. How can this benefit them? And how can I feel confident that it's really going to make a difference for them so that I should incorporate this into my practice? Because I think whether we're talking about Swiss providers or US providers, they care about their patients, right? They want them to have the best outcomes in a way that works best within the system. I also think that from the ethics perspective, the justice issue of being able to care for all of our patients across their racial and ethnic backgrounds and across socioeconomic boundaries is really, really critical, because I think, again, otherwise we're going to end up with multiple tiers of service delivery. It's already happening.
Yeah, thanks very much. And so I think one of the underlying issues here is in terms of articulating the message for the primary care providers and for the general public is, I wonder whether we should insist on educating the world for the new era of genomics, or if we should try to get rid of genetic exceptionalism and to just basically tell them this is not new. This is what we have been doing in medicine for years. If you go for a checkup when you're 40 and we discover a very high cholesterol, it's also a problem for your life insurance. It's also a problem for your future uncertainty, whatever. We just make it a bit larger, but it's the same thing. And those two approaches are almost mutually exclusive somehow. So who would like to say something about that?
Maybe I can start if okay, Marc. I mean, we did one study in Finland called Gene Risk, where we took middle-aged Finns. So this was a random population sample from southern Finland. We evaluated cardiovascular disease risks for everybody using both traditional methods as well as the best PRS at the time, this was 5 years ago, returned the information to the individuals themselves, and we have followed them up since. And one of the things we sort of spent a lot of time on was to ask them about how does it feel to receive this information? Does it worry you? Does it bother you? Does it motivate you? And then follow up what were the actions that those individuals who were in the high risk group, either because of their cholesterol levels or blood pressure or because of their PRS, what did they actually do and who responded to that? But when asking about, and then we had, multiple, we tried to be very careful in providing support to individuals. So we had hotlines, and we had, email and other many types of different ways that they could contact us if they were worried. And we got quite a lot of contacting from the individuals. This was 7,000 individuals, so there was quite a lot of phone calls. 98% of the phone calls were related on how do I get into this portal? What's my password? What's my username? How do I utilize this? Where is the information? And so, in other words, it's a very tiny fraction that, at least in our sample, of course, these were participants that decided to participate in this. So even if it's random sample originally, there's certainly selection of who comes, but there were very few people who were very much worried about the results. Some reported that they got very unexpected results. They may have gotten a low risk when they have multiple cases in their family, or the other way around. But still, so that tells, at least, in this small subsection of individuals, being Finns and being Southern Finland and so forth, I think it was really encouraging in terms of how, when you say that we need to teach the people, this is one way of teaching them, but making sure at the same time that we support those ones who are vulnerable or who are confused as much as possible.
Yeah, Marc, you wanted to add something to that topic?
Well, it's a very minor something more. To hinge on the sweet utopia of educating the public, which we should do, I think the PRSs have the added value that a polygenic problem is much less hereditary. And one of the difficulties in genetic counseling is what we'll do in your children, what will we do in your brothers and sisters when we find something in you, which is much more dimmer with the polygenic resource. So maybe very pragmatically, when people get used to it, and they will, it will a bit desacralize the very, I should say, abstract way that people are afraid of genetics in general.
Yeah, Kelly.
I just want to follow up on what Samuli said about the low number of people who have these strong responses. They absolutely do exist, but I completely agree with you. I think that we've created this structure of fear about genetic responses. And even for the highly penetrant Mendelian conditions, people may have a short-term response of some anxiety and depression and worry. And then if you can get them to that point of empowerment, they go right back to whatever their normal baseline is for how they exist in the world. And I think we have to also focus on that, this notion that we can empower people to have better health and make good choices, which I think actually seems to be quite important in Swiss culture. And if we can tap into that and have the supportive management things behind it, we really could make a big difference here.
So that's the optimistic view, but isn't it a real risk of ending up with very broad discrepancies? Because, only people already eating vegetables will be very interested in their genomes, to simplify the picture. So we end up with the low socioeconomic persons at the same place they are today, and some people that are already at advantage being at much better advantage than that, even before talking about human enhancement or whatever could come in coming decades. So is there a way to avoid that? And that's my main worry in the field, frankly.
Well, that's where we need the health psychology studies, right? About how we can better motivate people to make these behavior changes, and what we can do in our communication about the health issues that will help them somehow feel like they want to make that difference or take that step that they haven't otherwise taken.
Yes, Marc.
Yes, totally. And one way would be not to overprotect genetic data acquisition. Genetic diagnosis is just a diagnosis. If people knew what kind of incidental findings could pop up when you have a röntgenogram, a scanner of the abdomen because they have the pain here, maybe they wouldn't venture in it. But we don't present it that way, and they have a common sense which they don't have in genetics because it's so obscure to them. And I think we amplify this difficulty by overprotecting genetics as something, a totally weird animal, totally different from the rest of medicine, which it is not. And being very, I think we are all very much afraid of possible liabilities and other problems if we do not have very complex concepts. And if you go and do some query in the population about what we should do with the genetic data, it seems to be quite different from the ways that our ethical persons, or I should not say ethical, I should say the legal persons who draft the consents and our behavior think that we should do to be on the safe side. And probably it's something that should be diffused. And again, one way to diffuse it is start using the PRS, mentioning what it is and what it is not, as another piece of medicine which nobody understands anyway. I don't understand how a scanner works. I'm just happy to know what I have in my gallbladder, okay? And to some extent, the details will not help the patients to make the decision. What will help is what can come out of it and how can it be used for my health.
So I remind everyone that you're welcome to ask questions or comment on any of the topics using the Q&A or the chat features. Olivier, in oncology, we said it's used today more and more. Do you see a change in the way the patients are empowered, because they are at a moment in their life where they are confronted with a terrible disease? They were before the genomic era. Are they living through it differently because they have this additional layer of information, or it doesn't really matter to the deceased person with the cancer?
No, I think it does crucially impact on the way they want to be treated and the way they interact with the doctors. Now they are much better informed of the benefits of having some types of genomics analysis of their tumor, and we are frequently being asked to perform genomics analysis in some instance for patients that are relapsing, and sometimes it's justified, sometimes it's not, depending on what we can action after that. But clearly, I think the level of awareness of what this can be used for is really increasing in our patient, and we really do see this.
The Genome Center, which is hosting this event today, received clinical accreditation a couple of months ago, so we are very proud. And I want to congratulate the whole team that worked extremely hard over a pandemic year and more to get to that stage. So it is now possible to get your full genome, full transcriptome, full exome at the Genome Center with the ISO certification clinical grade, meaning that whenever you send a DNA or an RNA sample to be sequenced, you'll get clinical grade, even if it's for a research project, meaning that some of the hurdles that were present before are not there anymore. We don't have the excuse to say it's research grade only, we cannot use in the clinic. So my question now would be, is that a sea change for at least Western Switzerland or whole of Switzerland, meaning that we should push for large-scale cohorts to, for example, have the genetic data immediately incorporated into electronic health record to facilitate this transition? Or is it something that will be impossible to do considering the current context in the country? And that's maybe for our two Swiss practitioners, but also Kelly, because they started discovering the legal landscape.
Yeah, of course, it opens great possibilities, but the question will be who will pay for it. And usually now in the practice, if you want to have an exome or a genome for a hereditary disease, you need to have the insurance, the medical insurance provide the money. And so probably the cohort will depend on the indication. And probably also the authority will want to maintain the overall cost, changing the techniques that I use, but keep within the same budget and envelope. So probably we will not, I don't see an easy way that we will be able to build up cohorts and then design what we want to do with it once a new PRS or a new application pops up.
In oncology, we might have a different use case, which is the molecular tumor board and the precision oncology programs, where large panels are already being reimbursed in most cases by the insurance companies. And I think there is an opportunity to go much broader and to collect data. We have now databases to track the clinical evolution of those patients. So if we could have the genomics data at a larger scale, I think this would be very useful. Of course, the branching point will need to be pathology departments, because that's where the samples are being analyzed and the small scale panels being done. But we could see a case where, for some patients, larger panels or exomes or transcriptomics could be done at the center. And that, I think the fact that this is clinical grade is fantastic for a precision oncology program, because this would allow us to use that information for our decision making.
Yeah, I agree that clinical grade piece makes it a lot easier once you make the decision that you want to return some set of results. I fully agree with Marc's comments that the logistics and the money are going to be a real challenge. And you may want to think about how you can set up a system with some equity if you're going to begin returning these results. So for example, is there going to be, I know Effie said there wasn't a list of genes that was selected for return, but you probably don't want to end up in a situation where one set of research projects returns one set of genes and a different set of research projects returns a completely different set of conditions, unless it's only paired with the indication for research. So I would worry a bit about that, and then how things were getting into the medical records. For equity reasons, you don't want patients to have access to different things. And really then there's the research question of what is it RGB to actually provide to people, and how do we balance that with our desire to give them as much information as you have around things that we have some clinical validity and utility to.
Yeah, so one problem I always hear about bringing genomics to the people, especially the preventative medicine part, is that we don't have a system, at least in our country, that reimburse health. We have a disease insurance, not a health insurance in Switzerland. And by law, if I'm not mistaken, even if we proved tomorrow that sequencing the genome of every single Swiss citizen is cost efficient, it wouldn't be reimbursed because it's not for personal care. So should we stop everything we do and just go to march on Bern and lobby the parliamentarian until they create a new law, or is there a way around that? And here I'd be curious to hear about what's happening in north of Europe, Samuli. Do you think in your healthcare systems that you will be able to use PRS at the individual level? Could it be reimbursed by a basic universal insurance, or is that the same problem you have there as we have here?
I think we sort of a short answer is that we do have the same problem. It's a slightly different flavor of the same problem. For the primary prevention is sort of in the hands of the occupational healthcare, which for those individuals who are working. So, and there's a sort of insurance in the background, but that's run mostly by private healthcare providers in Finland. And the business model is that it's the employers who buy a package to their employees. So and then comes the fact that if they spend annually, I don't know, 100 euros per employee, you cannot put on top of that something which costs 500 euros. So that's where the economics are. So once the the cost of the sequencing comes down enough, then I think these will start to, be more implemented in the health. So that's the slightly longer term. I'm sure it's coming, but not quite yet.
Yeah, Marc?
I think, Jacques, you gave the hint to the answer of that question. Maybe one way to go would to be to find another source of reimbursement income, not from the disease system, as you mentioned, but from prevention. And I can tell you a story from my former country, Belgium, where there is a social security for disease, which is held by the federal government. And then there are budgets for prevention, which are under the control of the cantons, to make it simple. And genetics may fall between the two seats occasionally, and that was sometimes very surreal. But it's clearly very different to try to have precision medicine by better defining what are the molecular pathways in somebody who's already sick. And then to have prevention using scores or any kind of approach in someone who isn't, but may have some reason to be in a higher group. And so maybe the idea would be to find some money elsewhere. Because in medicine, if you want to get more money for genetics, someone will have to give away money from his budget. Maybe the gastroenterologist, maybe the OBGYN, but that will create war. And I don't think that any decision maker will dare to go there. So they will tell you, okay, if you need the money, find it in your own envelope. Even if you spare money outside of your envelope, find it in yours. I can make accounts between you guys. So that might be, I think, some difficulty.
Ioannis, you want to intervene?
Yeah, I'm a Swiss citizen, despite my name. I'd like to have my genome resequenced because I sequenced it on probably 14 technologies since the first Illumina and also Affymetrix chip that said it was genome-wide that Lena Peltonen had access to, and we were doing the first GWAS on this. So how much actually the citizens should actually influence that? Because on one side, if I take the cost of a genome now over the entire year of lifespan of an individual, which is around 80 years, I found it rather that dirt cheap to sequence it and to interpret it. So I would see this more, we have seen the recreational one on the West Coast of the US with all its hooplas and problems and reselling of things. And I guess Kelly knows much more about this than me. But somehow, when we have the advantage of having someone like Olivier Menzel that works on rare disease, where basically there are parents that are in dire needs to have places where they could do clinical sequencing. So it's trying to put back the tool, and perhaps that might be the move from the Genome Center, is to put back the Genome Center in the hand of the citizen, rather, and the patient that we are all, because we are all comorbid, as Samuli showed that with the PRS. So basically, putting that back in the hand of the people and say, are you ready to sequence your genomes and be interested to have that genome that could be used for medical use later on and be used in the same way as we use the COVID softwares that we have on our cell phone for tracing and testing, basically. So it's I don't know whether I'm completely clear, but bring back the stuff in the hands of the citizen rather than the hands of the people that have the knowledge or the clinicians. That would be my claim for this, to move out of this insurance people money. At the end of the day, we have money. We are in Switzerland, and we are most of the Swiss are in a good financial status. And the genome at 800 francs, which I think is going down, is not that expensive if you think about this. An entire life. It's less
Absolutely. And we might start with the patients' associations who have clout, who have political ear, and they would be very representative to go this way.
Yeah, the full shortcut will not be feasible because of the new law that was just approved and still requires that any sequencing, genotyping, whatever that has medical relevance goes through the professional. So there is no shortcut of the profession, but it might be possible to build a structure that offers that. Of course, if people have to pay, it will be inequalities, at least short term. But then maybe to rebound on what Marc said before about the two-level reimbursement scheme, in Switzerland, if I understand correctly, it's the same thing. So prevention is supposed to be cantonal matter. And there is maybe probably something to do with the stratification of screening that Samuli presented, because there is colorectal cancer screening, breast cancer screening paid by the cantons. And there is a clear case to be made to do the genome large scale to improve on that. And that could be a weak point where we should go full speed and try to convince some cantonal authorities to play the role of pioneers. Is anything like that happening? Do you know, Olivier, if you know in preventive oncology, people are talking about doing the genome stratified screening?
I mean, I know that there are, projects, but I don't know of any, nationwide idea to move this, and I think it would be a brilliant idea. I think also, of course, here we're talking about prevention, but patients that are affected by cancer are, really extremely keen on having anything tested. I mean, it's clear for them that any thing that slows down their access to care or drugs is really perceived very, and rightly so, very, very negatively. And so I think we have also here a leverage to make sure we can bring the correct message to the politician to ease some of this, because it's really very needed in different patient populations.
Okay, so maybe we'll go for last round of questions, unless there are other topics that are coming. We have talked a lot about what to do immediately. Where will we be in a few more years in terms of genomic influx into the healthcare system? So do you believe in this longitudinal multi-omics follow-up at the individual level, or is that a Silicon Valley dream for some high-tech person that will never say never, but that will not happen in the near or middle future, because it just won't have the delta necessary on the health of people to justify the investment and all the fanciness around it. So I think let's try to think 10 years, 15 years from now. Is that happening, or is that not happening at all? What do you think?
Maybe I can start. I think just I like the sort of the longitudinal multi-omics world, in a sense that I mean some of our main ingredients of our risk are what we eat and how we move. So regardless of our polygenic risk scores, we should eat more healthy and probably go jogging or running once or twice more per week, on average at least. So and then comes the behavioral changes related to preventative medicine, and how do we measure those? And I think that's where the omics and the different kind of trackers, different kind of new technologies offer us tremendous opportunities, since we all know how difficult it is to record your diet based on a questionnaire or physical activity as well. So they provide us opportunities to combine the background risks, the interventions, and the follow-up and outcome of these interventions in a very new way. And I think that's going to be very powerful also when we try to argue back that we should do these types of preventive interventions. So I think that's going to be an important one component of the next few years, on top of perhaps still we are going to have, now that you have the clinical grade sequencing, I think you will find a way to sequence everybody in Switzerland relatively soon. Maybe not this year, and maybe not even next year, but maybe after that.
Yes, Marc.
Yeah, absolutely. And one way to look at it may be penetrance. Multi-omics is a way to reintroduce part of the phenotype to increase the penetrance of the whole predictive test you are doing. Look at the current story. What is clearly working is monogenics and cancer. And we are both looking at highly penetrant changes, and that works. And the limit of the PRS, I think, in my opinion, and Samuli, you correct me if I'm wrong, has been twofold. One is that the full penetrance is usually around 50%. And the second was that PRS so far have been less performing in those 50%, but are now coming of age. Now you can uncover the 50%, but it still remains 50%. If you can feed in or mix, that is feeding what is missing, you will have a highly powerful tool. So although it is not pervasive in the clinic right now, if I should guess, yes, in the coming years it will.
Yeah, I think in oncology and for patients who are affected by the disease, I think it's almost a no discussion that longitudinal multiomics is the way to go. I think it's at this time precision oncology programs, but it's pretty clear that from that we'll identify the crucial elements that we will need to follow longitudinally in patients to really improve survival. So for me, it's pretty clear that that's going to be in the clinics ASAP.
Yeah, I agree that this is going to be a reality very soon. And I think that one of the things that we learned from COVID is that people are willing to use the mHealth applications, and they're willing to do things like genome sequencing if they feel trust. So I also think that we need to set up all of these processes in a way that really engender trust with the Swiss people so that they're willing to go all in and then be able to reap the benefits that we're talking about here.
I really like ending with that word trust, because that's the key. Trust among scientists, between us, like translational researchers, and the medical doctors that are really, for many of them, not really aware of what has been bubbling in the background, and of course, trust with the general population. Excellent. So, I don't know if any of the panelists wants to say something else. Otherwise, I think Manolis will say a few final words. And I want to thank you all, the audience, and of course our panelists, and again the speakers of today. It was a real pleasure to share this moment with you all. Manolis.
Okay. Well, thank you, Jacques. Thank you very much. I just want to say that this has been an amazing day. I think it was not only excellent science that was presented, but also forward-looking science, forward-looking ideas, and forward-looking concerns and issues that have come up. I think that contrary to many meetings that I've been in, sort of genomics in general, where it was basically just sort of a layout of just discovery after discovery of specific projects, I think that the issues that were tackled today were actually broader, and we went in many different directions: precision medicine, drug discovery, how this is done at the population level, aspects of ethics, and it was really a very nice collection of themes that honestly was probably much better than we had anticipated when we originally were thinking about the speakers. So thank you all for actually being so thoughtful about what you presented and for making this a success. We would have loved to have this in person. I think would have been even more exciting, and continue these exchanges over coffee or a few drinks and maybe a nice dinner afterwards. Unfortunately, these are not for the time being, but hopefully in the future. I just want to thank everyone, in particular Olivier Menzel, who organized all this and was responsible for the invitations, for the organization, for all the administrative parts. Everybody who chaired the sessions, primarily, of course, the speakers who presented, and of course the panel members, and every one of the attendees for actually investing their time to listen. And there's more to come, many exciting developments, and I'm sure the Genome Center will continue to be in the center of those developments in Switzerland and Europe. And we're all looking forward to see this developing over the next few years. So thank you very much again. Have a good weekend, and enjoy your dinner, even though you cannot have it all together.
Thanks.